afternoon, everyone. I'm Yan Li from Xi'an, China. Appreciated the conference gave me this opportunity to share our recent work. My topic is NDRG2, a new estrogen-targeted gene. The full name of this gene is NMYC downstream regulated gene 2, and it was first found and cloned in Dr. Yao's lab in our university in 1999. Recently, we checked the promoter of this gene. We found there is a potential ERE, estrogen response element, on the promoter of this gene. But this is not exactly like the classic one. One nucleotide is different. First, we want to know whether NDRG2 can be affected by estrogen. And we found both the transcriptional levels and the protein levels of NDRG2 can be upregulated by the estrogen with dose and time-dependent manners in HeLa cells. We further detect the whether this ERE exists on the promoter, and we designed the deletion mutants and point mutants. Finally, we find only the full length of the promoter can activate the luciferase activity, and the point mutant of G2T is crucial for the active of luciferase in the promoter system. There are two kinds of estrogen receptors. One is alpha receptor, the other one is beta receptor. The estrogen only bind with the receptors, then it can be activated transcriptional factor. We find in HEK2 nice red cell, these two cells that only the beta receptor can activate the luciferase, but not alpha receptor. We also apply the deletion mutant in exon 7 to 8 part. This part is crucial for the binding of a beta receptor and estrogen. And we find that in this mutant, the activity of the luciferase was decreased obviously. Next, we use 3 cell lines. They are MCF7, HSG, and T47D. These 3 cell lines are all both alpha receptor and beta receptor positive cells. We treatment with the cell with PPT and DPN. PPT is the alpha receptor specific agonist, and DPN is the beta receptor specific agonist. We find only the DPN can activate the report system in among the 3 cell lines. We further apply the siRNA to silence the expression of a beta receptor, and the luciferase activity was attenuated obviously. Next, we want to further confirm our results. So we applied 2 experiments. One is CHIP. This in 2 cell lines, and CHIP can show that the binding of the estrogen and the NDRG2 promoter, which is beta receptor dependent in vivo, we also in vitro levels with the EMSA experiment to show the binding is beta receptor dependent. Right now, we can got the conclusion that NDRH2 is a new estrogen-targeted gene. So the following question is, what's that mean? Whether it's related with some physiological or pathological functions. In fact, since 1999, a lot of paper publication show that NDRH2 is involved in the cell proliferation and the differentiation. And this gene also is a cell stress response gene. Interesting, NDRH2 also associated with some ions transport, the sodium transport through epithelial sodium channel. Through the tissue RNA dot hybridization analysis, we found NDRH2, its expression is really, this is a human tissues, and this part is salivary gland. It's very interesting. In salivary gland, this tissue, the expression of NDRH2 is relatively high. We further check the location of NDRH2 by immunohistochemistry analysis in 3 big pairs of human salivary glands and rats submandibular glands. We found NDRH2's expression is very specific, mainly expressed on the ductal cells in the salivary gland, both in human and rat tissues. But in the actin cells, its expression is relatively weak. So we revealed the structure of the salivary glands and the salivary secretion. The salivary gland contains 2 kinds of cells. It's actin cells and the ductal cells. In fact, the original saliva first secrete from the actin cells. That time, the concentration of the sodium is very high, and it's hypersonic, hypersonic. With the original saliva enter the small duct, the sodium was reabsorption, so the sodium changed to the isotonic. Then the saliva further into the big ducts, and the final saliva in our mouth is hypotonic. It's the concentration of the sodium is only 1/9 of the original saliva. So you will feel very comfortable, the saliva in your mouth. Oral dryness or xerostomia is a kind of subjective symptom, which is prevalent in the post-menopausal women. About 1/3 of the post-menopausal women suffered from the oral dryness. To mimic the situation of the post-menopause women, we remove the ovary of the rat and collect the saliva before and after the surgery. And we check the content of the saliva. We found the sodium and the chloride, the concentration increased when after the OVX surgery in the salivary, in the saliva. But the potassium and the calcium without too much change. We also found the saliva flow rate decreased in the OVX and animal model. And the OVX rats will drink more water From this data, we can got the conclusion that our animal model, the OVX animal, can successfully mimic the oral dryness, which is estrogen deficiency induced oral dryness. And we check the protein level in the OVX rats, in the salivary gland of the OVX rats. We found NDRG2, its protein level decreased by the histochemistry and the Western blot analysis. Another two important sodium transporters, sodium potassium ATPase and epithelial sodium channel, its protein level also decreased. But in the MR level, only NDRG2 and epithelial sodium channel, the levels decreased. It seems that the oral dryness induced by the estrogen deficiency is have some associated with decreased NDRG2. So next we want to rescue the NDRG2. We designed two adenovirus or adenovirus carry the NDRG2 gene. One is CMV promoter derived. The other one is KLL. CMV, this is non-specific, and KLL is tissue-specific, which can keep the NDRG2 expressed only in the ductal cell. After the OVX surgeon, we treatment the NDRG2 adenovirus through the retrograde ductal administration. 3 days later, we check the expression of NDRG2, find in the KL-derived adenovirus, NDRG2 expressed only in the ductal cell. The D shows the ductal cell, and in CMV-derived adenovirus, the NDRG2 expressed both in the acinar cells and ductal cells without specific. 3 days later, we also checked the concentration and other symptoms of the rats, and we found in the saliva both the sodium and the chlorhydrin decreased to the relatively normal level, and the saliva flow rate increased, restored to the physiological level, and the water intake of the animals decreased to the normal. This data showed that NDRG2 rescue may be a potential therapeutic or intervention to the oral dryness. Because of time, I skipped some slides, but I would like to explain it further through this work model. This is the ductal cell section. This part is the apical side. The opposite one is the basolateral side. In the normal level, estrogen combined with the beta receptor as an activated transcription factor to activate the expression of NDRG2. And NDRG2 can bind with the beta subunit of sodium potassium ATPase. And on the basolateral side, the sodium potassium ATPase can pump the sodium out of the cells. On the other side, the apical side, the epithelial sodium channel can sense decreased sodium, and so in the apical side, the sodium in the saliva can be reabsorbed into the cell. So both sides can keep the balance. On the other, on the post-menopause women, in these conditions, the decreased estrogen can't active the beta receptor, and the NDRG2's level decreased. Lost the protection of the NDRG2, the beta subunit of sodium potassium ATPase the beta subunit will be ubiquitinated and degraded through the UPS system. So the balance between these two sides can be broken. So in the apical side, the sodium can't be reabsorbed into the cell very well. So the concentration in the saliva will very high. So some people will feel the oral dryness, especially the postmenopause women. So I would like to thank very talented students in my lab, Li Shaoqing, Yang Jiandong, Liu Changhao, and my cooperation of other professors in our university. And I would like to thank Dr. Zheng from NIH and Gustav Fashion from Karolinska. Thank you for their give us provide the plasmids and other tools to finish our research, and thank the financial support from China. Thank you.
All right, folks, do we have any questions right now? Any questions? Okay.
Thank you. Very nice work. Congratulations. I would like, have you analyzed only the ENaC beta subunit in the alpha subunit? Have you analyzing this the expression or no? The ENaC, sodium epithelium channel. Have you analyzed the other subunits of the sodium channel?
No. We only check the beta subunit.
Only usually, yeah. I'm not asking just because usually the alpha subunit is higher modulated too, and
alpha, beta, and gamma. But the beta one is constitute subunits. So
now you analyze the beta subunit of sodium potassium ATPase, not a beta of ENaC. Okay, understand. Okay, and your proposal is that the NDRG2 directly interacts with the beta subunit of the sodium potassium ATPase and precludes its ubiquitination. That's it? That's what I understand. That your molecule...
Yes. In fact, I skipped some slides, so it's hard to understand.
Okay, precludes the...
we use hybridization analysis. Is East2 hybridization, and we found NDRG2 combined with beta subunit of sodium potassium.
Okay, you detected this interaction, direct interaction by the hybrid system.
Yeah, the mechanism part we published in GBC, and I haven't time to show that. So
okay, thank. And in kidney, do you have any? Because you didn't show kidney, just have
kidney.
Yeah.
The tissue. Yeah. In fact, my colleagues in his group checked not only the salivary. NDRG2 is very specific on the ductal cell, not only in the salivary gland, but also in the kidney and breast, all in the duct cell.
Hmm.
As the expression is very high.
In
And
collecting duct cell? No, I don't.
Duct cell.
Okay.
DUCTAL, duct cell.
Okay.
Mm.
And you have any change? Do you have any change in ENaC in renal tubules? No, you don't. In this.
Pardon?
When you have the expression of this protein in collecting duct in renal, collecting duct tubules, collecting duct in kidney. Do you have expression of this protein? In
the kidney.
Yeah. In principal cells.
Yeah, in my lab, we just focused on
okay.
salivary.
Thank you.
With the yeast to hybrid, it's sometimes necessary to prove the result with some other techniques such as colocalization and Western blot co-immunoprecipitation, something like that. Have you also tried that to confirm your result?
You mean when I rescue the endogenous expression?
No, after doing the yeast to hybrid assay, you have to prove the result with co-immunoprecipitation, some protein work such as colocalization and Western blot following co-immunoprecipitation. I'm just wondering, have you ever tried that also to prove? Because just the direct interaction in yeast to hybrid is not enough to confirm the interaction of two proteins.
Yes, we also do the co-immunic appreciation and the co-immunostaining. It skips too much information, sorry.
Also, you colocalize them in the cells?
Yes, in the cells.
Okay, good.